I have to confess, I'm an immunologist. I'm not a nanomedicine person. And what I'm going to talk to you about is a thesis project for a graduate student that turned into something that was a bigger fish, okay? So this paper has been published in PNAS, so I'm just going to try to go through this as efficiently as I can. So at least you understand my title, Vesicles as a Mediator of Microchimerism and Split Tolerance. What is microchimerism? When I talk about that, we are all born as chimeras. We live our whole lives as chimeras, and we're going to die as chimeras. Basically, a chimera is as Ray Owen called it originally, the man who discovered chimerism, incongruous juxtaposition. And it's epitomized by the statue in Arezzo, you know, the lion with the goat coming out of the middle and the snake on the tail. So we're all of that. And sometimes in the course of our lives, if one happens to have a uterus, one can also gain chimerism that way. If one gets a transplant, which is where my field, one can become a super chimera, that is to become more chimeric. So one of the things that happens, and what is our goal when we started this graduate student out on the project, was just to understand why it was. And then we published a paper 20 years ago about this. In kidney transplantation between siblings, when the mismatch is for mother, and that's that 6th chromosome which contains the MHC antigens, is so important in transplant. If you get the same chromosome, you and your sibling, from father, that means your sister or brother is going to look just like mother, right? Because that's the non-inherited antigen, which we call the Nima. And so the paradox of the Nima transplant, if you will, was that there was absolutely an increased rate of early onset acute rejections. 80% of these transplants were almost immediately had an acute rejection response. But the long term was a decreased rate of late graft loss. So what's causing this paradoxical effect? Okay, and so I'm basically summarize my talk right here, and you can cease to pay attention to me the rest of the way. The answer is maternal microchimerism, which we all have, produces exosomes. These exosomes modify our own DC, and the result is a DC that's doing two different things, okay, at the same time, causing split tolerance. So I have to push this a little hard. Okay, well, you guys have seen very good presentations already on people who are far more expert than I. This just to illustrate two of the aspects that the exosome does. One is, of course, it transfers information in the cargo of the microRNAs and the different kinds of RNA species. It also does this, which I will emphasize because I'm an immunologist. I'm terribly interested in this. And so these are the exosomes produced by dendritic cells. So dendritic cells are the main antigen-presenting cells of the body. They are rather very good at presenting to T cells the class II peptides of exogenously acquired and the class I peptides endogenously acquired. And they also, of course, have the tetraspanin marker. So this is these two are very important, but this one is also equally important. If you get the signal 1, that is the peptide, your T cell is not enough to activate them. They need another signal. And so this CD86 is the way that the dendritic cell passes the information. This is a positive. You must respond now to this, okay? So we'll talk about CD86 versus PDL1, and PDL1 as the negative signal that we think is coming from the cargo information here. So this is the model. Basically, we're crossing a homozygous B6 mouse, B by B, with a heterozygous mother. This means that the offspring that have B by B, like the father here, are actually different from father because they've been exposed to microchimerism. We call this MMC, maternal microchimerism, of the mother. This microchimerism does two very different things in about half of the mice. Half of the mice get only a soluble antigen that is not encoded in a vesicle. The other half of the mice who become tolerant to a transplant, the thing that we're interested in as transplanters, they get both the soluble antigen as well as exosome encoded antigens contained in exosomes. This term, we use the terms MAAQ for membrane alloantigen acquisition as the positive. However, this term has also been used cross-dressed. Don't ask me where that came from. It came from immunologists who are dealing with other issues. But I like to call this the mark of Zorro, but that's another thing. So anyway, basically my talk is to compare these two groups of mice, all right? And so I have various tools. I have antibody tools to detect the MHC class 1 KFD, the MHC class 2 IAFD, and I also have this wonderful tool. It's an antibody that does what the T cell does. The T cell's not looking at the whole molecule. It's really looking at that peptide in the groove. And this antibody is a very unique antibody because it does the same thing. So when I come to read out function, I'll talk about these two. This 4C cell recognizes the intact IAFD molecule with its peptides. And then this is very specific, like this antibody. It only recognizes one particular peptide. This gets the whole talk across. A picture's worth 1,000 words. Basically, can you tell which of these cells has been peppered with exosomes, right? Okay, so the mice that are not MAQ, they have CD11C, which is the marker for dendritic cells, but they don't have this peppered appearance, and that's because they have not been peppered with exosomes. So we're trying to figure out what is the difference here. What sorts of things are happening? Why is this happening, first of all? So we take the serum of the mouse, and we look at the serum with a very crude kind of ExoQuick type preparation, which many people in the field will turn their nose down at. But it actually does give you something that has a fairly good range of sizes. And most importantly, although it's not completely clean, you can see there's ER markers and there's histone. A large part of this is an exosome marker, that is the CD9. And so it's, let's say, enriched in exosomes. So we're going to call it EV or extracellular vesicles from here on. So what happens when you just take a B6 mouse, completely naive, that's not involved in one of these crossbreeding experiments, and you just say, okay, let me take those cells and incubate them with the exosome-enriched fraction of the EV of the MAQ mouse versus the non-MAQ mouse. So what you see here is that's the CD9, that's the exosome there. These are three that have attached to the MDC of the host. So this is MDC, by the way, stands for myeloid DC. PDCs are plasmacytoid. DCs are rather different cells, and they have different membrane fluidities. And you can see this one in 6 hours has already uptaked all of the antigen very nicely, whereas this one is rather slow. And what you see is basically, depending on the source, you get this phenomenon. You do not get this phenomenon in either MDC or the PDC if you use the MAQ negative as your source. One other thing I want to say about the serum of these mice, the non-MAQ mouse, that is the mouse that only gets the soluble form from mother, is not devoid of her antigen. In fact, is actually quite a good signal from the EV-free fraction, okay? So, but all of that signal from mother is in that EV-free fraction. There is no signal at all in the non-EV-free fraction, okay? And since this antigen here, i.e. is the substrate for the peptide that we're going to be looking at with the YE antibody, it's a very important fact that both fractions, EV and EV enriched and EV free, are present in the MAQ mice. So that's the story of serum. What about the story of the dendritic cells? This just kind of summarizes what we know about that. The MAQ cells have increased their PD-L1 level, okay? They have also decreased their antigen processing. So this YAE is actually the result of the processing of this. And as you see, these cells have processed very well, the non-MAQ mice, but these cells rather poorly. So you have two very, very different things going on. So, all right, so this is the crux of the matter. What one can do with this image stream type of analysis is you get, it's kind of like population microscopy. You're running a lot of cells, and instead of just getting a signal, you're getting an actual photograph of these cells as they go through. So this is a 60x photograph. And what you're seeing here is what we call bright field similarity. It tells you how close these two markers are on the surface of the cell. So in this case, when we compare where the class 1 of the mother and the class 2 of the mother, they're located in exactly the same spot. So these are, a 4 is about as close as you can be, according to this method. However, the PD-L1, which we saw was elevated, is not at all in that spot. In fact, if it was, you would see yellow here, because the green and the red would interfere. Instead, you have a lot of PD-L1 going on, but a very low similarity between them, so they're really separated. In contrast to that, the peptide, that is the peptide that's seen by the YAE antibody, you do see some yellow going on. So they're two different parts of the, basically you were looking at different parts of the same cell. And to summarize, one part is saying go, positive, and the other part is saying stop. Okay? So here's the sort of the diagram. And the soluble antigen, which we saw was high in the non-MAQ mice, right? That soluble antigen is feeding in here very nicely and is stimulating this cell, which we call TEA. On the other hand, the MAQ mice has, in addition, these vesicles, right? So the vesicles are peppering the surface of that cell, and they're forming an entity for the recognition by the what we call a semi-direct pathway, which confuses everyone, so I'm not going to go there. But it's basically, it's a different type of, it's looking for the intact protein from the exosome, okay? Whereas this is the broken down piece, right? All right, so here's what my graduate student finally did. He isolated the dendritic cells of these mice, okay? So the first thing he did was he analyzed the function with these T cells. How did they respond to the dendritic cells of the non-MAQ mouse? And as you can see, because there's no exosomes bringing the antigen for the 4C, it does not respond, okay? So this cell does not respond. However, the TEA cell responds very, very well to a mother's antigen because it's broken down rapidly and they're able to see it. And the addition of PD-L1 doesn't really do anything to that. In contrast, the MAQ in the mouse, which now has the exosomes and has those little punctate regions of its surface, that gives a nice stimulation to the 4C cell. But the TEA cell cannot respond. However, if you use an antibody to PD-L1, which of course we know is very big in tumor therapy now, to break that tolerance, voila, strong response to that stimulation. So this is the final, I hope I can wind it up quickly. In the non-MAQ situation, we have exosomes being made, but they're always being made by the host, only by the host, right? Mother is not making, that mother is maybe a tissue-resident cell that cannot produce or does not wish to produce these exosomes, and instead she's just producing this soluble form. This soluble form is getting broken down through the classic pathway, and it's being presented in a very strong CD86 context, and we get proliferation. And that's simply amplified by the exosome pathway of the internal cell. In contrast, oops, I don't want to go back. How do I go back? Can I go back? Yes. In the MAQ positive mouse, you have a very different thing going on because mother's not only making this, she's also making this. And so these vesicles have the ability to cause stimulation of the 4C cell, which was not stimulated over here at all. And so that's the positive. That context is in a CD86 zone of the surface. And we've shown that there's CD86 in those hotspots on the surface. But in contrast, the cell that's looking for the processed peptide, these are being, we think, functionally reprogrammed by RNA species. And so my last slide is just where we want to go. This is where I want to use nanoparticles. It's, for me, not medicine. It's to answer a question, all right? And so we have a collaborator, Shigwan Gan at Madison, and she is going to make a polykaryotic core, and she's going to put both microRNA candidates as well as the E-alpha peptide or full-length E into this nanoparticle. And I should say that we already have 3 that has been recently published. All 3 of these do 2 things. They increase PD-L1 and they interfere with antigen processing. So maybe there's many more out there, but we at least have a place to start looking. And I should acknowledge our collaborators, Jan van Rood, who was actually originally invited to give this talk by Beat, but fortunately I was able to come. Jan is a bit old. He's the age of Beat's dad, okay? And so he didn't want to come all this way. Franz Klaas also in Leiden, Todd Brennan at Duke, who provided us with a 4C cell, and Jean Florentin, who did the nice Western blot analysis, Partha Duda, who started. And this is the man who got us all. Everybody in my lab, just about, is now doing exosome work. And it's all his fault because he was doing this very simple project and it turned into an exosome project. So thanks very much.
Thank you very much. I saw Jon van der Rood about 5 years ago. I had no idea that he was that aged. So when I saw him on the paper, I thought, oh, that's an excellent candidate for a talk. But then he politely declined.
Yes.
Yes. Yeah. Questions? It's a bit prosaic, but how can we use this phenomenon for medical purposes, or?
Yeah, so I would say it's going to be useful in tolerance. If we, for instance, if we identify the microRNA that's really causing the other side, that side of the dendritic cell to push down processing and cause the PD-L1 kind of negative thing, then we can incorporate whatever we want. I mean, if someone is, a diabetic, insulin, we can maybe incorporate myelin basic proteins, things of that sort.
Is it really an underestimated player in the whole field of the immune system, basically, the extracellular vesicles? Do you feel that way? Yeah,
absolutely.
Is it the mediator?
This is why we're all working on it right now. And again, I was very interested in your talk and the idea that the dendritic cells talk to each other. So if we wanted to use those vesicles and load them, we could also be talking to the right cell. And that's the idea. But T cells seem to have another way of talking to each other through their own.
Questions from the audience? No? Then I do remind you that at 3 o'clock, somewhere outside, we will be discussing the extracellular vesicle field. I hope that all the people that have presented today will join me there and ask additional questions or be asked additional questions. At 3 o'clock, we'll be outside. I would like to thank all the speakers for their really nice talks and, well, talk to you soon. Thank you.